Hi, this is Lake Müggelsee, Berlin's biggest lake with a vital ecosystem. In 2011, the quagga mussel invaded this lake and since then disrupted the food web massively by filtering out phytoplankton, tiny microscopic organisms. The mussels form dense carpets underwater with up to 35,000 individuals per square meter. Every mussel can filter 300 milliliters per hour, so statistically the whole water of Lake Müggelsee is filtered roughly 1 to 2 times per day.
Quagga mussels often arrive in water bodies through human impacts, for example by boats or sports equipment. When they arrive, they reproduce fast and in high numbers, so quickly they colonize large parts of the sediment. In Lake Müggelsee we can find different types of cyanobacteria, tiny microorganisms that can produce toxins. There is Anabaena, Microcystis, which can aggregate and form colonies, Aphanizomenon, which can form filamentous aggregates, and there are the green algae Chlorella and later Cenedesmus. All of these belong to the group of phytoplankton, which form the base of the food web in lakes. Quagga mussels are filter feeders, which means they feed on removing particles such as phytoplankton from the water to metabolize them. Their feeding behavior has found to be selective, so they decide themselves which food they like. In our experiments we could find that green algae, here Chlorella and Cenedesmus, serve as good food. The same counts for Anabaena, whereas filtration on colonial and filamentous cyanobacteria, Microcystis and Aphanizomenon was lower. In the context of global warming, temperatures and especially heat waves in lakes are increasing. These higher temperatures are strongly affecting the filtration of phytoplankton by quagga mussels. Rising temperatures are stressing the mussels and reduce their activity. This in the end can lead to an increased occurrence of cyanobacteria and thus endanger the health of ecosystems and humans. So on many sunny days we went into the fields to Lake Müggelsee and collected mussels as well as phytoplankton. For some experiments we used plankton nets with a specific mesh size to increase the share of colonial microorganisms. This resulted in concentrated samples which resemble potentially harmful algal blooms. Under the microscope we can see the different morphologies of the organisms. In the lab we test the filtration of quagga mussels on different phytoplankton species from lab cultures as well as from the field in incubators on a temperature range from 24 to 32 degrees. The results will give us more information on how quagga mussels will shape harmful algal blooms in lakes under the stress of climate change in the future.